Dear students, welcome to this lecture. Today we'll be discussing structure of angiosperm, seed and embryo. Seed is considered as a wonder of nature. It's the defining feature of angiosperms. The seed consists of a miniature plant in the form of an embryo. Seed in a small package has got everything of a mature full-fledged plant, that is, root, shoot, leaves, source of nourishment and protective coverings. Seeds occur in gymnosperms also, but they are naked and are borne on sporophylls. Seeds in angiosperms are covered and develop inside the ovary of a flower. Seed is a special and highly adaptive structure. It's critical in the continuation of the flowering plant species. Evolution of seed habit has been a key factor in the migration of plants from aquatic to truly terrestrial habitats. Seed ensures a survival of plant through unfavorable and hostile environmental conditions. It perinates in the form of an inactive dormant propagule, suspending all activities of life temporarily during unfavorable times. At the time of return of suitable conditions, cellular activities are resumed, mostly on absorption of water from the surrounding atmosphere, leading to seed germination. Let us discuss the structures that make up a seed one by one. First is the seed coat. Seed coat is the outer covering of the seeds. It's protective in nature. The outer layer of seed coat is known as testa and the inner one as tegmen. In certain cases, like cereals, in addition to the seed coat, ovary wall also protects the embryo. The seed coat functions mainly to prevent the seed from drying and desiccation. It's usually thin and papery, as in bean and peanut, but it can be thick and hard or stony, as in case of Brazil nut. Seed coats are responsible for dormancy in many seeds. For example, Amaranthus retroflexus and in members of Malvaceae and Fabaceae, etc. The seed coat of the cotton plant produces cotton fibers, which are spun into thread and woven into a cloth. The seed coat has a mark of attachment of the stalk of ovule to the ovary wall, which is known as hilum. The micropyle, that's the opening enclosed by integuments of ovule, is also visible in some seeds as a tiny pore or a dark patch. Micropyle forms the point of entry of water into the seed at the time of germination. Next is the embryo. Embryo lies inside the seed coat. Embryo by far is the most important part of the seed. Other structures exist made to ensure its well-being, security and survival. Embryo is the miniature plant in actuality. It consists of embryonic root known as the radicle, embryonic shoot known as plumule and one or two embryonic leaves known as cotyledons. In a dicot seed such as bean, cotyledons form the most prominent part of the seed. The cotyledons function to store food and supply nourishment to the developing embryo until its own leaves become functional. The cotyledons of a monocot seed do not store nutrients. Rather, they absorb nutrients from the endosperm and transfer them to the embryo. The embryonic axis is known as tegellum. The part of tegellum that lies just below cotyledonary node is called the hypocotyle, and the part of this axis that lies just above this node is known as the epicotyle. Next structure in the seed is endosperm. Endosperm is a nutritive tissue. Endosperm stores starch, proteins, hemicelluloses, oils and fats. Some plant families like Trapaceae, Podocytamaceae and Orchidaceae are remarkable in lacking this tissue. Endosperm produces hostorial appendages which may be given out at callosal end or at micropylar end. Hostorial appendages may also be given out laterally. In several plants, hostorial appendages are given out at more than one places. These appendages serve to absorb the nutrition from the surrounding tissue to fulfill the additional nutritional requirements of the developing embryo. In cereal grains, the outermost layer of endosperm is known as aileron layer. The cells of aileron layer contain aileron grains which are bound by a unit membrane. Apart from being storehouse for important nutrients like proteins, phytin, phospholipids and carbohydrates, aileron grains produce hydrolytic enzymes like amylases and proteases during seed germination. Endosperm is formed after triple fusion. Therefore, it's triploid, that is 3N in most angiosperms. However, in Oinothera lamarckiana, it's diploid, as it's formed from the fusion of a male gamete with only a single polar nucleus. Embryo sac is only 4 nucleate in this plant. In Peperomia pellucida, it may be 15N. The highest polarity level has been reported for Adam maculatum, where it may be as high as 24,576N and is the haploid chromosome number. Now we'll talk about types of endosperm. Endosperm is of 3 types on the basis of its development. First is the nuclear endosperm, second is the cellular endosperm, and third, halobale endosperm. In case of nuclear endosperm, free nuclear divisions take place during endosperm formation and cell wall is not laid between these nuclei. In cellular endosperm, after triple fusion and formation of primary endosperm nucleus, nuclear divisions are followed by wall formation. With the result, the type of endosperm formed is cellular. In halobale endosperm, first division of primary endosperm nucleus results in the formation of a large micropylar cell and a small callosal cell. Nucleus of a micropylar cell divides freely and that of a callosal cell either remains undivided or divides only a few times. The callosal cell is also called as a basal apparatus. Halobale endosperm is mostly found in case of monocots. At the later stages of development, endosperm becomes cellular in all the cases. Nuclear endosperm is generally regarded as primitive and cellular endosperm as advanced, and halobale is considered intermediate between these two. However, some workers believe reverse to be true. In addition to these 3 types of endosperms, endosperm in some angiosperm families, which are approximately 32 in number, including Anonaceae, Apocynaceae, Aristolochiaceae, Caprifoliaceae, Ebinaceae, Meristicaceae, Palmae, Passifloraceae, Polygonaceae, and Rubiaceae, etc., has an uneven and irregular surface. Such an endosperm is known as ruminate endosperm. Ruminate is derived from the Latin word ruminatus, which means chived. Rumination is believed to arise from ingrowths or infoldings of seed coat or by unequal radial elongation of seed coat cells. Ruminate endosperm is further divided into 7 types on the basis of its morphology. This was about the types of endosperm. Now we'll talk about types of seeds. Seeds in angiosperms are categorized on the basis of criteria including number first, number of cotyledons in the seed, and number second, presence or absence of endosperm. The classification of seeds on the basis of a number of cotyledons. Angiosperms are differentiated into 2 important groups on the basis of a number of cotyledons in the seed. Number first is the monocots and number second dicots. This categorization is supported by many other characters which are very distinct in these 2 groups of plants. Now first, the monocot or monocotyledon seed. Monocot seeds possess only a single cotyledon that does not store nutrients. Corn is a typical example. Although a corn grain is usually a fruit, but the seed occupies almost the entire grain. The wall of the fruit, known as a pericarp, is fused to the seed coat. Below this lies the aileron layer, which is composed of thick-walled aileron cells. Endosperm occurs below the aileron layer. The single shield-shaped cotyledon, known as scutellum, lies on the upper side of the embryo. An epithelial layer separates scutellum from the endosperm. Near the base of the grain lies the embryo, which is placed laterally. At the lower end of embryonic axis lies the radicle, which remains covered by a sheath known as coleorhiza. The upper part of the embryonic axis, that is phylumule, is also surrounded by a sheath which is known as coleoptile. Now the dicot or dicotyledon seed. Dicots have two cotyledons in their seed. A typical dicot seed is exemplified by pea seed. Pea seed is covered by seed coat consisting of outer thick testa and inner thin tegmen. Seed coat carries the funicular scar, that is hilum. Micropyle is placed very close to hilum. Embryo or a kernel becomes visible after the seed coat is removed. The embryo is clearly distinguishable into tegellum with radicle at the lower end and phylumule at the upper end. Two fleshy cotyledons are laterally attached to the tegellum. Now the classification of seeds on the basis of presence or absence of endosperm. Seeds are categorized into two types on the basis of presence or absence of endosperm. These are endospermic seeds, which are also known as albuminous seeds, and non-endospermic seeds, also known as ex-albuminous seeds. Endospermic seeds or albuminous seeds retain endosperm even after maturity. Examples include Ricinus, Tamarindus, Zea, and Oryza, etc. In non-endospermic seeds or ex-albuminous seeds, endosperm is not retained after maturity as all of it is consumed during the development of seeds. Examples include Fasciolus, Cicer, Vallisneria, and Elisma. In addition to endosperm, some seeds retain a thin layer of nuclear tissue. Such seeds are called as endospermic perispermic or simply perispermic seeds. For example, zingiber, eleuteria, and curcuma, etc. The edible part of a coffee seed is perisperm actually. Now the development of seed. Seeds develop after fertilization of ovules. An ovule is an integumented megaspore angium. Structurally, it consists of a stalk known as a funicle and a filling tissue, nucellus, covered by one or two cover layers, the integuments. The place where funicle joins the ovule is known as chalaza. At the opposite end, the integuments enclose an opening which is known as a micropyle. At maturity, a megaspore mother cell is differentiated from the rest of nucellar cells in the ovule. Megaspore mother cell gives rise to megaspores after meiosis, and megaspores give rise to embryo sac after variable number of post-meiotic mitotic divisions. The number of post-meiotic mitotic divisions is fixed for different plant species. On the basis of number of megaspores involved in the formation of embryo sac are of three types: monosporic, bisporic, and tetrasporic. In case of monosporic embryo sacs, only one megaspore is involved in the development of embryo sac, and in bisporic embryo sacs, a dyad is involved in embryo sac formation. Monosporic and bisporic embryo sacs are further categorized depending on whether micropylar or a chalazal megaspore or megaspore dyad gives rise to mature embryo sac. In case of tetrasporic embryo sacs, all the four megaspores formed after meiosis in megaspore mother cell contribute towards the development of mature embryo sac. A mature embryo sac in most of the angiosperms has an 8-nucleate and 7-celled organization, including 3-celled egg apparatus at the micropylar end, which consists of an egg and two synergids on its sides, two polar nuclei which may fuse to form the primary endosperm nucleus, and three antipodals at the chalazae end. An embryo sac is also known as a female gametophyte in angiosperms. Pollen tube delivers male gametes to the female gametophyte. The nucleus of one of the male gametes fuses with the egg and that of the other with the primary endosperm nucleus. The former, that's a fusion of one of the male nuclei with egg, is known as fertilization or syngamy, and later, since it involves union of 3 nuclei in most of the angiosperms, is known as a triple fusion. Syngamy was first discovered by Strasburger in 1884 in Monotropa. While syngamy leads to the formation of embryo, triple fusion results in the formation of nutritive tissue endosperm. Syngamy and triple fusion constitute what is commonly known as double fertilization, a phenomena unique to angiosperms. Now we'll talk briefly about the seed dispersal. The fundamental purpose of a seed is to serve as a means of reproduction in plants. However, since higher plants lack locomotion, seed dispersal assumes a special significance. Seeds represent an excellent means of dispersal of plants to far off places. Seeds are dispersed mostly when still inside fruits. However, some seeds aid in their own dispersal. Different agencies are involved in seed dispersal. These include wind, water, animals, ants, etc. Now first the wind. Dispersal by wind is known as anemochory. Anemochorous seeds are usually light in weight, and some seeds have developed hairs or wings to facilitate their dispersal by wind. Lightweight is the most common adaptation for anemochory. Examples are orchard seeds. The seeds in some plants like Calotropis, Allostonia, Gossypium, Hollerina, etc. have hairs that help them float in the air. Such seeds are described as comos. Hairy seeds also occur in poplars. In several members of Asteraceae, for example Taraxacum, Tragopogon, etc., Dipsaceae and Valerianaceae, the calyx is modified into hairy pappus. It helps the fruit, which contains the seed, to float in the air. Seeds as well as fruits in many plants develop wings to facilitate dispersal by wind. Winged seeds are found in Alsomitra macrocarpa. Next dispersal agency is water. Dispersal by water is known as hydrochory. Plants growing on a seashore or within water disperse their seeds and fruits by hydrochory. Plants adapted to hydrochorous seed dispersal produce buoyant seeds which are carried by water currents and are washed ashore at far off places. For example, seeds of Alisma, Nymphea, Sagittaria, Nilambo, etc. Seeds of Cocos, Erica and Nipa, etc. are also dispersed by water. The seeds of mangrove plants float on the water surface for months. Next dispersal agency is animals. Dispersal by animals is known as zoochory. Such seeds have hooks or barbs on their seed coat which attach them to the fur, feather or skin of animals, aiding in their dispersal. Hooks form an important adaptation for zoochory. Plant seeds that have hooks to effect zoochory include Xanthium, Articum, Medicago, Geum, Emex, Pedalium, Tribulus, etc. In Rafflesia and Dendrophtho, seeds have a sticky testa that gets attached to the body of animals which aid in their dispersal. While the seeds of Rafflesia are dispersed by elephants, those of Dendrophtho are dispersed by birds. Next is the ants. Dispersal of seeds by ants is known as myrmecochory. Foraging ants disperse seeds which have appendages called elaiosomes. Elaiosomes are soft fleshy structures that contain nutrients for the animals that eat them. Elaiosomes are eaten by ants and the rest of the seed is discarded by them because of being hard and inedible. Such kind of dispersal is seen in case of acacia and trillium, etc. Next is the self-dispersal. Some plants do not involve external agencies in the dispersal of their seeds. Instead, they produce explosive fruits which burst open forcefully, therefore dispersing the seeds. Such a mechanism of dispersal is known as autochory. The fruits of impatiens, oxalis and viola explode and twist on being touched. Similarly, the fruits of clitoria, abrus, bohenia and geranium burst open suddenly. Seeds in Rhelia, Barleria and Andrographis, etc. are located on small projections called jaculators or retinaculae. Jaculators push out the seeds with a jerk to effect their local dispersal. Now we'll talk about seed dormancy very briefly. Dormancy is the inability of seeds to germinate under conditions favorable for germination. Dormancy is very important as it allows dispersal of seeds and avoids precarious germination. The stalling of germination also safeguards seeds and seedlings from suffering damage or death, which may be caused due to short period of favorable weather followed by long periods of inclement and unfavorable weather. Dormancy also protects seeds from transient herbivores. Dormancy is usually categorized into two types: innate dormancy and enforced dormancy, depending on whether it results from environmental factors or from the conditions within the seed. Dormancy due to external factors is also known as imposed dormancy or acquiescence. Now, the causes of dormancy. While the imposed dormancy may arise due to unfavorable environmental conditions like unavailability of water, unfavorable temperature, or a clogged supply of oxygen, innate or true dormancy in a seed may be caused due to the following factors. Number first, resistant seed coat. For example, in plants like Amaranthus retroflexus, the seed coat is very hard and does not allow the radicle to pierce through, causing dormancy in the seeds. Number second, it can be due to impermeability of seed coat to water. Water is indispensable for germination. The seed coat in many members of Malvaceae, Fabaceae, Chenopodiaceae, Convolvulaceae and Solanaceae are impermeable to water, which inhibits germination. Seed coat impermeable to gases. The seed coat of plants like Xanthium is permeable to water but impermeable to oxygen. This causes a dormancy in seed as oxygen is required for mobilization of stored food products present in endosperm or in cotyledons. Number fourth, it can be due to rudimentary embryo. In some members of Orobranchaceae, Orchidaceae, as also in Ginkgo biloba, Fraxinus excelsior, Alex ephaeca, Eranthes, and Ranunculus species, seeds are shed at a stage when embryo is immature. Hence, the germination of seed is postponed till the embryo development is complete. Next, it can be due to dormant embryo. Embryo may be physiologically dormant at the time of shedding of seeds. Such seeds usually require a period commonly known as after ripening. After the period of after ripening is complete, seeds germinate normally. Dormancy for after ripening is seen in case of Pyrus, Prunus, Iris, Tulipa, Populus, etc. Germination inhibitors can be the other reason. Some chemical compounds like coumarins, para-ascorbic acids, ammonia, ferrolic acid and abscisic acid inhibit the plant growth in general and germination of seeds in particular. These natural germination inhibitors occur in seed coat, endosperm and embryo as well as in fruit pulp. Besides, cyanide releasing substances, phenolic compounds, organic acids, alkaloids and oils also inhibit seed germination. For a seed to germinate, leaching of these inhibitors or their sequestration is required. Non-dormant seeds may gain dormancy if they are kept under unfavorable conditions for a long time. Such dormancy is known as secondary dormancy. Now, seed germination. Germination is the process of coming out of seedling from the seed. Germination occurs when seed is not dormant and all conditions for its growth, external as well as internal, are satisfied. Under proper conditions of moisture, water, oxygen and temperature, seeds resume physiological functions and become active. At the time of germination, a radicle is the first to come out. It protrudes out through the micropyle and establishes contact with surrounding soil particles and develops into root. Pilumule comes out later and develops into shoot. Water is the primary requirement for germination. The next essential requirement is oxygen, though germination can proceed for some time even in absence of oxygen. Another important requirement for seed germination is optimum temperature. Seeds germinate over a wide range of temperature, usually between 16 to 25 degrees Celsius. However, optimum temperature varies for different plant species. Light is required only in case of positively photoblastic seeds to germinate. Among the internal factors for germination, viability of seed is the most important one, as only viable seeds germinate. Different seeds are viable for different periods of time. The longest viability has been reported for Nelumbo speciosa, which is about 800 years. Completion of dormancy is essential before seed germination can begin. Seed germination is usually of two types, apigeal germination and hypogeal germination. When the hypocotyle elongates quickly, bringing the cotyledons in the air, the germination is said to be apigeal. Apigeal germination is seen in many dicots, for example, castor, tamarind, gourds, and rarely in monocots, for example, onion. In hypogeal germination, the epicotyle elongates quickly, leaving the cotyledons on the ground or inside the soil. Hypogeal germination is seen in many dicots like pea, gram, and in most of the monocots. With this account of structure of seed and structure of embryo in angiosperms, I conclude the present lecture. Hope you understood and enjoyed it.